Hi, folks, and welcome to our next Connected Conservation, our C2 webinar in our series. My name is Cass Bismeyer, and I'm with the National Park Service. Thanks so much for tuning in today. Before we get to our webinar, just a little bit about our Connected Conservation Community of Practice. We're a team that is interdisciplinary, built out of lots of different folks from across the National Park Service in parks, regions, and our Washington office. Our focus is on working at the land and seascape scale and encouraging parks and programs to work beyond their boundaries. And so we've plugged a couple links into the chat there where you can learn a little bit more about C2 and take a look at some of the resources that we have available there as well. And as a part of our work, we offer a bi-monthly webinar series where we touch on topics that are working beyond the boundaries. We showcase examples where we're doing really well at this. We feature policies that enable us to do this type of work, among other aspects of working at the landscape level. All of the webinars are recorded, so we'll record this one and post this one as well. You can find them on both of our internal resource site as well as our external resource site. So with that said, again, thanks so much for tuning in. Feel free to plug in questions into that chat function throughout. No need to wait until the end. After we have our presentation, we'll wrap it up with a Q&A session and address those questions as time allows. With that said, I'm really honored to introduce Dr. Kristen Hart today. She is joining us from the U.S. Geological Survey. She is a research ecologist in the Wetland and Aquatic Research Center, and she's going to talk to us a little bit about her work in tracking marine turtles across the seascape. Thanks again, Kristen, and it's all yours.
Thank you so much, and thanks for joining today. I'm excited to talk about some of the work I do and have been doing for about 12 to 13 years. And so some of these photos are from our field work in the dry Tortugas, which is one of the places that's near and dear to my heart, and where I'll be headed tomorrow. So thanks for joining. Long-legged marine turtles have complex life cycles, often using spatially separate breeding and foraging grounds. These areas might be hundreds to thousands of kilometers apart. An extensive use of satellite and GPS-telemetry of turtles tagged on nesting beaches has shown pathways used by those individuals to transit from important breeding areas to foraging sites where they remain resident. Locations of migratory corridors have also emerged. And so this is a tool, a key tool that is really revealing high use zones in the marine environment, which are critically important for these imperiled sea turtles. In recent years, tracking of marine turtles that nest in U.S. national parks and protected areas has been a priority for understanding their spatial habitat use patterns. Real-time online tracking like that through the Animal Telemetry Network has illuminated connections among parks that were previously unknown. And this is where a really exciting aspect of my own work has come in to play. Throughout this presentation, I'll show some individual tracks, but also I'll start noting some areas where we're finding that consistently pop up for different species of turtles and single species of turtles as important beyond our national parks. A lot of the work I do is within national parks for the original tagging, but then where they go after that brief encounter with us is very important to understand. So these are just some of the pictures of green turtles and loggerheads and hawksbills in our various study sites. I'm always amazed by the number of times I have to briefly explain this sort of system to get data. But basically, it's a lot of work to tag and track these turtles, but we get them in the water and on land, we tag them. The tags actually transmit through the Earth orbiting satellites and then back down to us with a certain number of messages, and we get a location, and we can plot those locations. So this is just a really brief sort of magical slide where it all happens and we get data every day once we tag turtles. And so today I wanted to focus on some sea turtle connections that we're starting to see across the seascape in my own project. And I put this map together a long time ago, but it is pretty instructive. We work in the Virgin Islands, and some of those turtles go to the Bahamas. We work in the dry Tortugas in Southwest Florida at the end of the Florida Reef Tract, and some of those go to the Bahamas. So there's these like meeting points in the sea. Then some of our animals go north, and I also worked on the Deepwater Horizon oil spill, so you'll see some of those animals. But there's connections within the Gulf And then we also have a really interesting group of animals that go to Mexico. There's one more that isn't on this map, but she connects striped tortugas all the way down to Nicaragua. So some of these maps are within, or because we work in the national parks, we now have sort of interesting areas at sea that are popping up as really important to connect these individuals. Today I'll be talking about some land-based work and some in-water work, and these are just some of the photos we have of our crew at work. We do spend a lot of nights up all night. A lot of what I do is trying to decipher animal movement and behavior. So we're looking at animal movement tracks. There's specific locations in which they're spending time or doing interesting behaviors. We have actually involved some accelerometers in our tracking work, so we can not only know where they're going, but what they're doing. And then recently, I would say within the last 8 or 9 years, we've been really able to decipher more animal behavior from satellite tracks themselves with switching state space modeling, which I'll mention. And then we do things like quantify site loyalty. How long are turtles in a certain location, of a certain size? How long do they stay put? And those are critical information parameters for managers as they start managing place-based rules and things like that that would affect sea turtles. The standard data we collect is sightings and captures per unit surveyed. It allows us to estimate sort of a catch per unit effort, abundance, and density. We do we collect unique mark recapture events because we get our hands on every individual. That allows you to estimate survival, abundance, population growth rates, and habitat use. We take measurements of everybody, genetic samples, diet samples, and blood samples. So everyone is fully worked up. The analytical part I'll briefly mention, but with the location data in particular, we take the raw data, we do switching state space modeling to identify periods of transiting or periods of what we call area restricted search, so we're wandering. And then we identify those as migration, the transiting, really direct movement versus a foraging kind of mode. We filter data, quantify the site fidelity, and do statistical comparisons. And this figure at the top just shows like clustered points of Alabama, then this animal moved and migrated and settled down on the Florida shelf in the blue points. The study sites today I'll be talking about are these, Bon Secours National Wildlife Refuge, where I worked during oil spill, Everglades and dry tortugas here in South Florida, and then Buck Island Reef National Monument down in St. Croix. The first one I wanted to talk about was Everglades because it's in my backyard. So this is the first map, just shows our steady site in a little star. It's the remote area called Cape Sable. And it is actually a very large rookery for loggerheads, but it's logistically very difficult to get to and work in. So we have worked there since 2014. And the beaches are, quote unquote mainland beaches, but they might not operate just like the real mainland Florida beaches because it really is quite a remote site. We're primarily tagging loggerheads there that, although I have seen at least 1 or 2 nests of green turtles in the area. So this is what the smattering of points looks like for the 33 animals we've tagged so far. Some stay pretty close, which is interesting. Loggerheads are supposed to move. And some actually do make fairly large migrations out of the area. And you can see we do have international movements of these animals. The next site is one of my favorite sites, which is dry tortugas, so I'll spend a few more minutes on this. This is about 80 to 100 miles west of Key West, and it's accessible only by boat or seaplane. So it's really remote, it's out there, and it's a really marine park. There are a lot of small little islands, and you can see it in this map. It's not meant to show detail, but the right half of the park, the eastern half of the park, has fewer restrictions than the sort of pie-shaped left part of the park, where there's rules about anchoring, boating, things like that. And so a lot of what we also do is in-water work, which is really fun. This is a male sea turtle, and you don't get your hands on those unless you get in the water. So in this project, we're calling it 3.5 species. We have loggerheads, which are a genetically distinct subpopulation, green turtles, hawksbills, and then hybrid loggerhead hawksbills, which is kind of interesting. We have several capture methods, as I mentioned. We're catching them on the nesting beach at night and then in the water with dip net, turtle jumping or rodeo, and hand captures. And these are just some of the pictures that we have. And that's my 26-foot boat. That is our main research vessel. and some of the photos of us catching turtles, and then programming satellite tags on the beach. Our workhorse has been the middle one for a long time. The middle tag is from Wildlife Computers. It's a spot, but recently they upgraded it and they call it a rhino model now, which has a more protected status for the antenna and a few more bells and whistles. And we're seeing really great success with this. But we've deployed 175 satellite tags on animals out there, 48 of them on turtles captured in the water. So we have more than nesting females, which is really exciting. We do have females in the water, but we also have subadults and males and a few juveniles. So this is what that project actually looks like over all time. And it's fun to show this, but then what's interesting is we have over 40,000 turtle tracking days. A lot of them stay in the Florida Keys National Marine Sanctuary, which is in South Florida in the Keys. We have a range of tracking that goes up to 2,205 days. On one turtle that is resident in the Tortugas. That's a male. We've published a lot of our results from this particular project recently, but this one is the realized track migration of marine turtles, and this is a little photo of how we put satellite tags on, and this is important because we actually get really good retention times. And this is a foraging site map. The blue dots are study sites, but this is kind of almost my entire tracking data set, minus the last year or so, with where they go. And so you can see these clustered areas actually start popping up as common areas, and I'll bring this to the end. I'll bring the same type of stuff up, but we have common areas where these animals go to the Bahamas. Some stay in the Keys, some go north, some go over to Mexico, Nicaragua, and then the Buck Island turtles off to the right in the Caribbean are mostly from Buck. Some of the work I wanted to focus on is available in our USGS professional page link, which will be shared. But basically, for the green turtle story in the dry Tortugas, we actually found that the turtles were using local and regional marine protected areas, including those in the dry Tortugas, like they're not leaving, which was interesting. So this is the map of the South Florida protected areas. This is Biscayne National Park on the right, Everglades National Park, Key West National Wildlife Refuge, the box in the middle, and then the Tortugas, which has a Tortugas North Ecological Reserve, the Tortugas South, which are those rectangular boxes, and then the whole thing is within the Florida Keys National Marine Sanctuary, which abuts Everglades National Park. And this is a map that shows where the green turtle females went after the breeding season. So you can see some remained in the Tortugas, some went to the Key West National Wildlife Refuge, and some scooted almost all the way up to Biscayne National Park and just outside Everglades National Park. So we're starting to make connections with animals, you know, on the green turtle side of things, really close to our other DOI lands. This is an additional figure that just shows the tracks of them and a few more turtles added. The next story is about loggerheads. A lot of them actually leave and go to the Bahamas, which we did not know until we started working there and tracking them. The big figure on the right actually shows our turtles tracked in purple, and those are just the center point of their home range once they reach the Bahamas. And all the other green ones, the darker, brighter green, are other people's studies, turtles that I got from either the literature or the coordinates from the authors themselves. So this is an interesting mix of places where not only the ones I tag, but animals tagged also in other places go at sea. And the tracks from this paper in the left figure, those are just their state-based model tracks. The foraging site fidelity that we've been able to quantify by tagging individuals in successive nesting years. So I might have seen them 2 years ago, and then I put another tag on a subset of individuals. It's been very interesting. We've been able to compare their paths and how similar they were in different years, and that's this figure on the left. And then also how close are they once they do settle down. And it's been very interesting. At a poor scale, I can say these animals are sometimes like a half a kilometer from their previous point that they settled on. So they really are taking up a home range in a very distinct, almost territory type area. And this is pervasive around loggerheads that we're tracking in all studies. Okay, so then I wanted to speak a little bit about the oil spill. The reason I actually went up to the Bon Secours National Wildlife Refuge was because of the Deepwater Horizon oil spill in 2010. So by 2011, we had a pretty serious effort tagging and sampling these females up there. And we covered an 18-mile stretch of beach near Gulf Shores, Alabama. I worked very closely with Meg Lamont. She is my colleague at USGS, and together we've been able to really figure out a fair bit about what these northern Gulf sea turtles are doing. So this work is representative of our 2011 to 2015 tracking, and these are the sites that we worked in. Meg and her employees worked in Eglin and St. Joe Peninsula, and we worked in Gulf Shores. And this is what the basic tracking map of these ladies looks like. And so again, some remained within the northern Gulf, but some also came south, and some settled almost in the dry Tortugas. So we've connected some of our study sites this way. Some went, very few went west, and then down into Mexico. We actually had a few more tracked right across the Gulf, so it's an interesting smattering of where they go and what they do. Meg and I have published with co-authors on 2 key papers. One of them is focused on the internesting period, so the time during the breeding season, and then the other one focuses on where they go outside of that and where they end up settling. This is where key internesting areas are. The red are animals tagged in Alabama, the yellow are the ones at Eglin, and the green dots are the ones that Meg has tagged at St. Joe. So this is during the breeding season, which is not terribly surprising, but they're near the nesting beach on which we tagged them during the breeding season. But we also had some go back and forth, which was interesting, between our 2 sites, which was really unheard of at the time. It falls very much in line with work our colleague, Dr. Brian Shamblin, is leading to do genetic mark recapture in the Northern Gulf, or the Northern Recovery Unit, which is in the Atlantic for Loggerheads. But we're hopefully going to be embarking upon that in the Northern Gulf very soon. They're resident foraging areas for these animals that we've tagged with the oil spill imposed on there. We're across the Gulf. The ones tagged in Alabama are in purple, the ones in St. Joe are green, and the ones in Eglin are orange. And so you can again see there's these interesting smattering of points once they leave the breeding area. And again, we have some points right outside dry Tortugas and in the Florida Keys Sanctuary. Okay, then another favorite project. In 2011, we started a project in St. Croix where we were working on in-water and land-based captures of hawksbills and green turtles. We were doing tangle netting, dip netting, and mostly hand capture, which ended up being free diving. Our capping methods there were actually focused on a huge acoustic network as well as satellite telemetry. And again, we did standard sampling for every individual for body condition, genetics, diet, and health. So in St. Croix, nesting hawksbills and green turtles have been at Buck Island for a long time, but green turtles are really dramatically increasing there. And this is what the animals look like. We had over 10,000 tracking days in a shorter project compared to the Tortugas, but we did have individuals lasting several years. And again, you can see Chulu went up to the Bahamas. There's a couple more that did, and then some crossed the Caribbean Sea and end up right off Nicaragua's coast. We published these in a few key papers. The green turtles are all in one, so you can see they're on the left-hand side. There's a map that shows the centroids of where they spend during the breeding season, which is pretty close to Buck Island. And that's the top map, and then the bottom map actually shows where they go after the breeding season, which is not that far away. So both from the Tortugas and Buck Island, I'm seeing animals almost not being migratory, which is very interesting and important for park management because they potentially can affect the animal in its entire lifespan as an adult if it remains resident. Important in-water foraging areas for the USGI adult female green turtles that left were in these various areas. And I have a star in the Caribbean one on the bottom right, because this is an animal that actually went to another national park. It's a Las Roques National Park, so it's not a US park, but it's off Venezuela. We did have the ones in panel A, the top right, that stayed near Buck Island, and some that went to the other side of St. Croix, which is Sandy Point, another protected area, and then other ones that went to St. Kitts, Nevis, and Antigua. So the Hawksville story is a little different, and the animals that we tagged there, we had more of a focus on hawksbills because they're critically endangered, and it is critical habitat at Buck Island for these animals. Most of them during internesting did stay within the U.S. National Park boundary, which is all these black dots over here. The black line shows the park boundary. And then what's interesting after that, though, is these really are international travelers, just like the Loggerheads and the Tortugas. A lot of these went outside through other countries, EEZs, and ended up well outside of any type of protected area. And that's a key message for conservation for hawksbills especially. So the common foraging areas, as you stand back and take a 30,000 level view of this, start to pop out as Bahamas, Nicaragua, several places in Florida, Yucatan Peninsula in Mexico, and Nicaragua. And so some of the work that we started to do when we were realizing this was species specific. And so in 2012, we published a paper about common areas for loggerheads in the Gulf of Mexico from multiple distinct nesting subpopulations. And so this is what that type of map looks like. And our colleagues from University of Florida and Moat participated in this work, but we were able to characterize these foraging site characteristics that are important for loggerheads in the long term. They select relatively shallow water, average of 58.5 kilometers from shore, with net primary productivity ranging up to from 250 to 1180 milligrams of chlorophyll per meter squared per day, and a certain sea surface temperature range. And this became important as we went further and we saw additional areas. So one in the Big Bend and Florida Keys, it was another area where we have common or shared areas for loggerheads from multiple distinct nesting subpopulations. This one has animals in the Big Bend of Florida, from dry Tortugas, Everglades, and Gulf Shores, and right in the area where Meg works, the St. Joe Peninsula. But we took a step further with our colleague Donna Shaver. We're putting these data sets together for multiple species. And this is where I feel like we really have a lot of conservation bang for your buck. We can start putting these multiple huge spatial data sets together and say, aha, there's areas where camps and loggerheads overlap at their foraging grounds. Those pop out as key places for additional either preservation or work in the water. And so this is a paper that we put together with all these different study sites on the bottom, which include camps and loggerhead tagging. And your eye is drawn to the top figure, which shows where we have overlap of hotspots where these animals are living. So they're resident foraging areas. There's definitely concern about this given the recent hurricane right through there. But the number of animals overlapping here was pretty striking. The bottom right figure actually shows the number of loggerhead subpopulations in dark blue. So 2 or more, and then the red shows the species overlap. So you can pretty much see where we have multiple species interacting. Donna and I with colleagues took this further to actually do some predicting and using sort of ensemble niche modeling to actually look for a larger swath of using our tracking data, where could these animals be occurring? And this paper is better in color, but it shows where the 2 species potentially could occur. And this is important for environmental impact assessments and things like that. So the spatial tracking data really has a lot of uses. I wanted to close and sort of show very specific turtles that are connecting the parks. One that I just mentioned, Cape Hatteras is definitely connected to dry Tortugas via this animal that we tracked starting in June. She nested on Cape Hatteras, and then she appears to be settling just on the dry Tortugas northern park boundary right now. So this is an exciting turtle to be watching. One turtle from the dry Tortugas, Miranda, we tagged her in May 2013. She's a very small turtle, so I'm not sure if that was her first nest, but she actually lives in the Everglades. So she lives at this White Star and nests out in the dry Tortuga. So she actually connects Everglades and Tortugas. Fatima is a turtle that nests in the Everglades, but lives in the dry Tortugas. So we have them going back and forth, which is really interesting. She also made an Atlantic foray at one point. Courtney is one that I mentioned before, but this turtle connected the dry Tortugas to Nicaragua, which we hadn't seen before. So I don't know what proportion of these females do that, but this is her long circuitous track down there. And she's one that didn't reach her foraging ground for a long time. So I was watching this every day, and she's very close to a protected area, if not within it, in Nicaragua. Shuli actually has a paper about her, but she's a Buck Island Reef Hawksbill that lives in the Bahamas. And we tracked her for a long period of time. So we actually tracked her to her foraging area in the Bahamas and then all the way back to Buck Island, where she actually nested not on Buck Island, but on the mainland. And then she returned to the Bahamas to the same exact foraging area, and we tracked her there. So we're getting a sense of how good site fidelity is, how good is their site fidelity compass over the years, which has drastic implications for understanding mark recapture data for these animals. This is the publication, if you're interested, that shows more details of her movements. And then Diana is another turtle that we tracked from the Bahamas all the way up, or from Buck Island all the way up to the Bahamas. And she is in a very similar spot compared to the dry Tortugas females. So I imagine they're different species, but they're in a very similar spot, which is interesting. I put this together with my help of Andrew Crowder, my employee, just to talk about the number of satellite tags that we deploy per project. The numbers in parentheses are the satellite tags deployed. So sometimes, like in the Virgin Islands, when we get that random loggerhead, we have a tag on hand and we're able to look at where it's going. We did get one Kemp's Ridley in Alabama, named it Donna, things like that. So this is just a pretty impressive number to date, and there will be more. I have a lot of people to thank for the logistics support, funding, Everglades, dry Tortugas. They've all been phenomenal, especially our Park Service counterparts, and lots of volunteers and permitting and funding agencies and things like that. I guess I can take questions.
Thanks so much, Kristen. And what an incredible opportunity for you and your whole team to be out in these ecosystems, and just I can only imagine every day out in the field must just be amazing. And I think it goes without saying, your last few slides there, just seeing how important these critical connections are between these different protected areas and the corridor protection along the way, and just all those pieces are so important to turtles species. We do have a few questions that are already in the queue. So folks, if you have questions, go ahead and plug them in there and we'll just go through and hit as many as we can. The first question we have here is, are the hybrid Loggerhead Hawksbill turtles able to breed with either parent species?
Yes, I believe so. We did actually get the nest of at least one of them, and the success rate was not great. And so one of the ways we were able to look even further into the genetics of these animals is to genotype the hatchlings. So I believe the answer is yes. And it's sort of, in a one or two to say how these hybrid babies do. I don't think they did as well as a standard Loggerhead or Hawksbill nest, but yeah, they seem to be doing okay. There's also a lot in Brazil that are hybrids, which is interesting to me. There's a big proportion of logger hawks or hawk heads. I don't know what we call them.
Our next question here, do you look at turtle populations in Northeast New York Harbor? We see turtle boat propeller deaths. Here at our park, any literature, signs, or wayside text published by the National Park Service to inform the public?
So I don't work up there. I haven't actually had a lot of boat strikes, except in our recent work in Biscayne National Park. We actually feel like we have a pretty high number that have prop scars on them. Some in the Tortugas do, but it's a very low proportion. I could certainly dig up some more literature on the proportion of boat strikes and things like that. There are certain projects right now happening where some of my colleagues are trying to enumerate boat strike probabilities and things like that based on animal behavior. So maybe some of that can help. And I could certainly dig up a few articles like that.
Great, yeah, and if there are follow-up questions that we're not able to answer right now, we can go ahead and plug those questions into the chat box and we can follow up with you directly after. So for instance, for that one, Kristen, I can put you in touch with the person who asked that. Another question here, I'm curious why leatherbacks aren't a focal species of this work. Is it abundance or regulations related to ESA, or both, or neither?
So we have been trained with nesting leatherbacks because those are low likelihood of occurring in our study sites, but we know how to tag them, and I don't have the training to put satellite tags on them. That's a specific, it's sort of a different carapace. So I basically focus on the hard shell turtles. It's not to say that we can't do leatherback work, and in fact, I would love to do more in-water leatherback work in the Gulf. But my colleague Chris Sasso is doing a lot of that, and then Kara Dodge from the New England Aquarium. So I think we could, but it would be in collaboration. You do need a specific boat to catch them because they're so big. You kind of scoop them out of the sea. We don't have that boat, but we certainly could do more. And I know some of the areas that we've designated as important areas for hard-shelled turtles would definitely be on the radar for either foraging areas for leatherbacks or even transiting areas. So we'd love to do more. It would take a little more training and a little more money and a little more boat. So that's sort of why that. And they're just very rare in my world where I work and when I've been working in those places.
Okay. Another question here. How long do the satellite tags remain on the turtles?
So the longest I have is over 2,200 days. That's on a male loggerhead in the Tortugas. I actually tagged him when I was 8 months pregnant. My daughter is 6 today. So we have a paper, that the one that I mentioned from 2021, it's in movement ecology, and we actually go through and project out why tags last as long as they do. But we get well over a year on most tags. It's interesting. It depends on species, but it also really depends on where you're tracking them. The fouling rates on sea turtles in the Northern Gulf are very high. And so the turtle may still have the tag, but it might not be transmitting because it's got fouling over the sensors. I'm really confident in my epoxy. That's why we actually put our epoxy protocol in that paper. I have had turtles come back 5 years later with the tag still on them. I've had turtles come back 3 years later and the tag is not there. So it seems to be either the turtle has knocked it off because of their behavior. I'm pretty confident that I've stuck it on there really well. We've even had green turtles going for a year and a half. So greens seem to be the hardest to keep a tag on. I feel like it has something to do with their diet and they're sort of more oily, but I do have very good luck tagging them for many of them for over a year. The battery in the tag says somewhere around 875 days or something for the standard model if you program it kind of in a basic way. So you can get that much time, but there are caveats. So yeah, I would point you to the movement ecology paper to read some of the reasons why tags don't last as long in certain areas and on certain species. But yeah, I'm surprised when they only last 50 days. And I'm again shocked that we have 2,200 and 10 days on Jackson, this male turtle. Pretty exciting.
Another question here. Observed whether climate change could be affecting sea turtle foraging and breeding behavior.
Yeah, so one of the things I hope to do in the future with longer battery life and tags getting better is track animals, like females, back from the breeding areas or from the foraging areas to the breeding areas. And we have maybe 25 that we've been able to do this with. And the timing of that is really interesting. We sort of don't know what that is. Donna has a good number. I have a good number. And we're starting to look at that. Like, when do they actually leave the nesting beach to get back to these areas? And so I think climate change definitely, if it's going to manifest itself in changes to the foraging areas or to their reproductive status, it might shift it earlier. It might shift it just not, according to how it's been. And so it's really key, I think, to be tracking them beyond just the, get away from just tracking females from the beach to the foraging areas, but start tagging them more at the foraging areas and tracking them back and looking at the timing of that stuff. We've been able to tag, and I'm really excited about this, over 43 male sea turtles, and we're writing a paper, hoping to finish that up. But it's about, of those that leave, when do they leave? Like, when are the males making breeding migrations? Because it's different timing than the females. And this is also, we don't know that much about when and where they mate. It's presumed that they mate either at the foraging areas or along the way. But we've been able to track males. Like in early spring last year during the pandemic, I watched a male leave from Key West. He came all the way up the reef track, all the way to Delray Beach down here. And then he spent about 3 to 4 weeks here, and then he went all the way back down. I don't know if he was following a female, if he mated with a female, off Key West and was sort of escorting her up. I don't know. He didn't seem to stop and have clustered locations all along the way. Maybe he mated off the nesting beach. Like, that's the kind of stuff that's really exciting to be able to figure out with the additional type of tag I mentioned, the accelerometer tag, or observations, or both. But to know when to be at those sites to observe that is really key information. So hopefully we can design studies in the future to do that based on the information we're getting now.
And a few more questions coming in. And again, folks, if you do have questions, feel free to log them into the chat. Are the high use foraging areas associated with areas with no commercial fishing, or places where regulations reduce mortality from commercial fishing?
Some of the high use areas that I know best are in the Florida Keys Sanctuary, and I actually don't have like fishing effort for grid cell. So that would be something we need to get our hands on somehow. There are new layers coming available where, imagine a boat has actually like a satellite tracker on it, and so you can make like boat high use areas and see if they coincide with the turtle high use areas. But not everyone agrees to have a tracker on their boat for reasons you might imagine. So we have to rely on non-spatial data sometimes. So it's not a perfect, easy analysis to do. There's certainly areas designated for fishing, but I don't necessarily have the effort data that goes in there. Robert Hardy put together a paper about fishing on the southwest Florida shelf, and it showed there was definitely loggerhead overlap. And I know they're hooked and things like that. So commercial fishing and sea turtle interactions is definitely a thing. We have a lot of shrimping in the Gulf, and they're shrimping in areas that are high use zones for turtles. So there's definitely overlap. There's work to be done, I think, to really quantify the extent of it and the timing of it. So I think there's more data layers that are probably necessary to really, really answer that question.
And then just, we'll just wrap up this with a last question about satellite tagging. And then if we didn't get to your questions today, we'll make sure that Kristen has a chance to follow up with you or has the information too. Couple of questions about satellite tagging that have come in and about the opportunity to collaborate with other organizations who also are doing satellite tagging, like the Sea Turtle Conservancy. And then sort of a secondary question, if you're working with any other parks in South Florida through your satellite tagging and other parks that are taking place.
Yeah, we're working with Long Key. We're out in the Marquesas, and so we're not just focused on federal parks, but it is, we're interested in state parks as well. But from our mission sort of area perspective, any DOI lands are kind of top priority. So national wildlife refuges, national parks, national seashores, things like that, are kind of easier, I guess, to get funding to work in because it fits our mandate to provide the science to guide management in those places. We are definitely open to collaboration. In fact, in the Wildlife Computers portal, I have a button where I can click share tags with partners, and that makes it really easy. If they also are tracking animals, we can put them together. Otherwise, we just end up sending files back and forth and sharing it. I have one analysis right now that we're doing where we've engaged the folks who have rehab turtles, and we're comparing them to wild turtles that we've tagged. So I think that'll be really instructive going in the future, whatever we find, to say how do they behave, or do they behave similarly, when and how. So yeah, we're definitely open for collaboration. We've got a lot of the methods and stuff figured out. My time is not free, so we often do have to associate our projects with a fund source. But we apply for grants all the time, inside and outside of USGS, and we're fairly successful with that. Yeah, so we're definitely willing to collaborate and share. And if like particular turtles are helpful for someone else's analysis, yeah, I mean, I've contributed 100 turtles to this global analysis that's not led by me, it's led by a colleague, and it just made sense because they're looking at the mismatch in home range scale between animals and then management structures, which is really important to consider.
Well, this has been really great, Kristen. Thank you so much for all your time and your work in putting together this presentation. We know you have a busy schedule and that you're out in the field as much as you can be. So thank you so much again for showcasing your work and really highlighting that value of corridors and the importance of them protecting them for species like turtles. Folks, we are working on our fall series, and our fall series is focused on corridors. And so stay tuned for our dates. They should be released, as well as topics. They should be released for the rest of the fall into December, sometime in the next week or so. You'll find access to register to the upcoming webinars on both of those websites that I had mentioned at the beginning. I'll plug those in the chat again in case you missed them. But thanks so much again for tuning in. We really appreciate everyone's time today. And then we did record the webinar today too. So if you know of a colleague who would benefit, whether they're within the National Park Service or outside, all of our series is open to the public. So watch for the recording to be posted on our YouTube channel, likely sometime later today. So thanks again. And thanks again, Kristen.
Yeah, thank you.
Yep. Bye-bye.
Bye.